So hi everyone, hi Oliver here, Microbehunter here. Hello and welcome to another Saturday live stream. As every week, I would like to welcome you to another microscopy session. And usually I do have to check some of the setup first, whether everything works. I do see myself also online, but I think my face is a little bit too bright. So let's turn down the light a little bit. I think it's otherwise this one is too bright. So I'm going to turn down the light intensity a little bit. Sound also seems to be okay. I'm just here. That's great. Thank you again for joining in. Today I would like to do three things specifically, as you probably have already seen in the thumbnail and in the announcement, and maybe some of you have also seen in a video which you uploaded just today, and it's about mosquitoes. Right, and I would like to give you today a little bit more background information and take a little bit more time. So what I have here under my stereo microscope are some mosquito larvae that are collected. Now, if you did not see my video yet, you can see it, of course, in this channel. I just made a short video where I basically show you how I put those mosquito larvae under the microscope and so on. But in this live stream, I'm going to take a little bit more time. So that's the first thing that I would like to do. The second thing that I would like to do is I would like I've got some look here, this here, are the leaves of an oak tree. As a matter of fact, these are the leaves of the oldest oak tree in Europe. It's a 1,000-year-old oak tree which I visited a couple of weeks ago, and I took these leaves along. Unfortunately, they're now completely dried out, but yeah. And I'm going to show you how to make the stomates, the openings of the leaf visible using a glue method. And then the third thing, a little bit unexpected, I'm going to show this to you as well. And look what I got here. Isn't this beautiful? I have to open it up for... Ooh, there's so many mosquitoes and flies and everything in here. Unbelievable. Look what I got here. Isn't this beautiful? Some rotten cherries. And I'm going to put the mold also under the microscope because I can see some interesting, beautiful sporangia here. Or fruit, or rather the fruiting bodies. That's what I should say. The fruiting bodies of the fungus here are visible already which make the spores. So that is the thing that I would like to put under the microscope as well. So I have not done this yet. So it's going to be a little bit of a new experiment for me as well, because I did not check those cherries yet. And and what I would like to do now is, like every time, I would like to, let's switch on the stereo microscope again. I would like to go through some of the first comments already. And if you're new to this live stream, if you're new to this format, you can ask me anything that you would like to ask me in the chat. I recommend that you click on select live chat, because then you get the chat comments in the correct order. And if you have a specific question for me, then please indicate this by saying @Oliver or @Microbehunter, because there's so many comments. Hello all around the world, to Lebanon, to Italy, to Belgium, okay, to London, the Netherlands, to Germany, okay. From Nicaragua, even from California. Okay, and the question here is what is your first question today? Is what is your favorite thing to look at under the microscope? I do not have specifically any particular favorite thing that I would like to look at. I'm very broad in that sense. For those of you who don't know me in my real life, I'm a biology teacher, and therefore I'm fascinated by observing nature. Generally, but if you ask me about and so I like many things, and different things have different fascinations. So, for example, those mosquito larvae here are kind of fun to look because they wiggle around. However, the plant cross sections I think are very beautiful. So it's more artistic in that sense. Yeah? So I do not have a specific thing that I would like to look at, but rather I have relatively broad interests. And what I'd like to do also is I'd like to kind of mix stereo microscopy together with compound microscopy. I mean, there are a couple of microorganisms and specimens that are a little bit more fun to look at. But if you've seen them already many times, then sometimes they don't become so interesting anymore, right? And then I would like to observe different things, and then they become interesting because they're simply new to observe. Yeah. So this is basically, so also again, hello from Germany. Okay. As well. So what I would like to Yeah, another question: do you still activate new accounts on the forum? That's a question. It's yes, I do that. However, unfortunately, I've received so many spam registrations on the forum, so that I cannot distinguish anymore what is spam and what's not spam. So if you want me to activate you, register first. Please answer the question, but also send me an email. If you send me an email, I'll definitely prioritize that. But these days with the artificial intelligence, it's able to also answer the questions. So the security questions are not as safe anymore. There's a question of why you want to join the forum, and usually I hope that people are going to say something have certain microscopes or certain specific interests, but nowadays AI is able to answer these questions as well. And some months ago I had so many spam comments that I nearly had to close down the forum, right? And I don't want that. And ever since I'm a little bit more strict, it's actually much better. But I do understand that some people are kind of waiting very long, and I would like to request, please do send me an email, and then I'll definitely unlock you very quickly. Okay, so that's the thing. Yeah. How long have I been teaching biology? Since 1999. So that's 25 years, and I studied microbiology at the university. So I've got a research background. I changed over into education 25 years ago, and have found out that teaching biology, it can also be sometimes a little bit theoretical. And therefore I decided some time ago to do microscopy YouTube and live streams and so on, because this kind of reconnects me a little bit with nature and with observation. Yeah, something that is a little bit lost sometimes when you're teaching biology. It can become very theoretical, right? We talk about theoretical stuff, cell division, the stages of mitosis, maybe certain anatomical aspects. How does the human heart work? Very theoretical, but not very hands-on. And this is also one of the reasons why some of my students, also former students, I was just recently told they occasionally start to join in and do a little bit of nature observation with me, because it kind of reconnects me a little bit with the reason why I became a biologist in the first place, which is essentially a fascination for living things. Okay. I will, for those of you who know the format, what I will do is I will occasionally simply interrupt the live stream to answer some of the questions. So if I did not get around answering your questions yet, please be patient. I will do that, but I want to keep the live stream moving a little bit. So first of all, where did I collect those larvae here? First of all, those mosquito larvae, that's not a good sign. I have to say that, right? Usually where there is standing water, and I collected this from my garden where there was some standing water from watering the plants. After a very short time, mosquitoes will lay their eggs in there, and then those larvae will develop. And it's not a good sign because I would say maybe not so much in Central Europe where I am right now, but mainly in tropical regions. Mosquitoes are not only a nuisance, but actually they can also transmit diseases. For this reason, you should try to avoid to have standing water, also because you just don't want to get bitten by mosquitoes, right? And what these larvae do is like all insects, the mosquitoes undergo so-called metamorphosis. This basically means that a female mosquito, after mating, will lay the eggs in the water, and those larvae will hatch. And then they will feed on detritus, which is small microscopic particles, decomposing material, bacteria. They will grow in size, and then they will form a pupa. And what you have over here, this large thing over here seems to be a pupa. And this means it stops moving, and it will actually form into a mosquito. And then after some time, the mosquito will come out and it will sit on the surface of the water. It will wait until its wings and its body starts to dry sufficiently, and then it will start to fly away. Immediately try to find some food, basically blood, because making eggs does consume a lot of energy, and therefore they immediately will try to look for food, right? They will mate, and then the cycle continues. And the reason, and they need water for reproduction because those larvae live in water, and therefore this reason, if you want to avoid mosquitoes, you have to kind of try to avoid standing water. This is, of course, not always easy, and especially these days with flooding and so on, this can actually cause some populations of mosquitoes to explode. right? Yeah, why is this relevant? Because mosquitoes, of course, can transmit more in the tropical regions, can transmit diseases like malaria and a whole range of other diseases. Yeah, and for this reason, yeah, you try to, of course, yeah, reduce the reproduction of those mosquitoes. As a matter of fact, I read that the Center of Disease Control in the United States is considering mosquitoes to be the most dangerous animal in the world. Most dangerous because there are estimates that around 1 million people die because of yeah, mosquito-borne diseases, including, of course, a lot of malaria, but not only. So these guys are interesting and fun to look at, but actually they can be a problem. It could also be that yeah. Also, yes, due to climate change and so on, there are now more and more invasive species as well. So it is possible that we can expect also the distribution and population of mosquitoes to kind of change, yeah, as with climate change, and not only climate change maybe, but also simply because of increased precipitation and rain and so on. Yeah? So this is just something I just want to share. And what I'm going to do then now is I'm also going to take a few of those mosquitoes and I'm going to show you how I put them under the compound microscope, because under the compound microscope they actually look even a little bit more interesting. But yeah, next question. I'm interested in collecting some vintage and antique microscopes. Are there any makers to look out for, especially for antiques? Now, I'm not an expert in antique microscopes, but I do know that there are people who actually do collect them. I probably, would say that the company Zeiss was quite yeah, revolutionary back in the day with vintage microscopes. Many of those vintage microscopes, you know that they're vintage because they're made of brass and copper. They have this yellowish metal, look very beautiful. And I'm not saying that these are the only manufacturers, but I just know that Zeiss back in the day in the 19th century, they were quite important in actually driving microscopy forward, because they of course also yeah, cooperated yeah, with the company Schott, I think, which is a glass making company with lenses. So yeah, there was quite a bit of stuff going on back in the 19th century. So that's the only thing that I know. But I'm not saying that these are the only ones. Okay, so actually I'm not an expert concerning vintage microscopes so much. Yeah. I bought the Swift 380T. I was playing with an HDMI camera. I don't know why. If I connect the camera to the eyepiece of the image, it's way darker than what I get from the trinocular. Well, first of all, what you can do is you can maybe try to adjust the brightness of the HDMI camera. That's one thing. And another thing that is always worth saying is that the human eyes indeed are very sensitive to light. Okay, so the dynamic range of the human eye is much larger than the one of a camera. So I wouldn't be surprised yeah, if it is, yeah. But generally it shouldn't be darker because the camera or the software or the driver should compensate that. So what I have over here right now is also an HDMI camera. Right, and another thing is that yeah, I would probably play around a little bit with the exposure time here. Okay, yeah, is something related to the optical? Yeah, is it something related to the optical path? I don't. Honestly, if there were something in the optical path, then honestly, I think you wouldn't be able to see anything at all. Okay, yeah. Can I use dark field on those larvae? Okay, I will try to do that. Okay, when I transfer it, please remind me. So what I'm going to do is, otherwise people start to become impatient. I would like to now, yeah, simply pick out some of these larvae. And yes, I know that I'm kind of repeating a little bit that what I've already done in my other video. So if you want to see a more better edited video, I would like to refer you to the video that I uploaded just recently. So, but I'm going to switch over now to the desk view. The desk view. Okay, here we go. By the way, there's a mosquito in here as well, which I'm going to show you later. Okay, so that's the desk view, and here are the mosquitoes, larvae. Okay. So, oh yeah, I already removed the lid, and now we have the following problem, and that is that those larvae are fairly large, fairly thick. So if I basically put a drop on here, I could do that, and you know what? I could try. I'm just going to show you for the sake of completion, but just so that Look, I've got some microscope slides here which have an indentation. These are concave. So this basically means that they're able to take larger samples, and when I put a cover glass on top, they don't quite crush as easily. So what I have to do here now is I have to kind of take those larvae, put them on here, and of course I could try to look at them at low magnification without a cover glass. Okay, so that's indeed something that I'm going to try first, or put it in here. However, there is a disadvantage with those slides, the concave slides, and that is that sometimes it acts a little bit like a lens, and it kind of does not give you always a very good image. And especially with certain objects, sometimes it's better if they're kind of pressed flat a little bit to kind of get a better depth of field. Okay, so we just have to try. Yeah. So what I'm going to do first is with this one over here. And here we have the following problem, that if I put the larvae on here and a cover glass on top, it's going to be crushed. And I don't mind so much it being crushed. But essentially this, if I completely block its movement, then it's also not fun to look at. So what I therefore do is the following. I take two cover glasses. I put one here on the left side and one on the right side, and they are supposed to be like spacers. Okay, maybe you've already seen some of my previous videos where I used my slide ringing table to make spacer rings. That's also possible. But I would say that this is kind of the easiest, most straightforward way. And now I have to do the following. I'm taking some tweezers and, not tweezers, a pipette. These are disposable pipettes. And look, what I've done is I cut off the tip to make it a little bit wider. So I'm able to kind of suck up the larvae a little bit more easily, because actually it would be much too thin for the larvae to go in. Yeah. And now I have to try it a little bit with good luck, because my eyesight is not quite good anymore either. And I would like to maybe grab a smaller one. Okay, yeah, I got one over here. And I put it on, I'm going to put some water back because it's floating right. I think there are two of them even. Bloop, I put it on here. Okay, this is one or two? I think it's one. Yeah. And it's kind of in here right now. And what I do is if I just allow the thing to kind of touch the edge of the cover glass, then you might see that the water is being drawn in, and this kind of fixes the cover glass to the slide. It's almost like due to capillary action. Let me put this away again here. And then what I recommend is, you have to see over here, this guy is kind of going a little bit here. Sometimes I might want to remove a little bit some of the water here. And you know what I'm going to do now? I'm just going to put it directly under the microscope without cover glass, just for the sake of trying it out. Okay? And we're going to see a little bit how this looks like. The scope view. Let's turn up the light. So this is, I have to flip out the condenser. The color, the rainbow colors that you see is because I've got DIC, which is polarized light. And so sometimes those colors look nice and I leave them just for being colorful. But most of you are going to see it in regular bright field. Yeah. I'm going to now put the thing here. And let's see what I'm able to see here. So it's not in focus at all. I don't even know where it is. Yeah. So let's see. Let's put it into focus. Okay, and here it is, right? Okay, so that's regular bright field. What you see is the line that you see on the left side. That is the edge of the cover glass. Yeah. No. Of the spacer cover glass. I did not put any cover glass on top. Okay. Yeah. So you see that it's kind of moving around. Actually, because we're staying at low magnification, we could just leave it like that. Yeah, because the cover glass wouldn't make a huge difference. Yeah? And you just can see that, I'm just using now the cover glass on the side kind of only to limit its movement, and it's wiggling around, but sometimes you don't want that because maybe you would like to observe something in more detail. And here it is. Yeah? With the polarized light, it looks like this. Okay, that's also the thing that I kind of used in my video and this kind of adds a little bit of color and not necessary, but just colors is sometimes nice. So there has now been the request to put the whole thing in dark field, and as a matter of fact, I do have dark field only for the low power. And look what I'm doing. I have to just change the filter around, here, and then open it up. But it's not dark field either. Ah, no, I have to do it like this. Here we go. This is not dark field. Yeah, actually, I could have. I totally didn't think about this. I could have actually used this dark field footage as well for for my other video that I published, but I totally forgot about that. Yeah. But actually, it looks also quite nice. Yeah. So as we look at the beauty of this mosquito larva, I would like to read again some of the comments that you've written, and just to respond to some of the comments as far as I can respond. Yeah, can you please use dark field on those larvae? That's what I'm doing right now. I just want to see how the relatively big microbes look in dark field. Yeah. Strictly speaking, I know that I've also been guilty of this, but strictly speaking, these are not microorganisms, even though you see it much better with a microscope. The reason why they're not microorganisms is because they are multicellular. I know it's a little bit of a question of definition, but normally we talk about single-celled organisms as being microorganisms. Okay, so it's kind of a little bit of a strange thing, because also tardigrades, for example, they're definitely microscopic. You need a microscope to see it, but per definition, strictly speaking, they're not microorganisms. Yeah? So it's you see it in biology. It's all a little bit sometimes conflicting definitions here. Yeah. Greetings from Sri Lanka. What do the larvae eat? What they eat is they eat so-called detritus. Apparently they're not very specific. Essentially, they eat pretty much anything that fits into the digestive system. Detritus means decomposing plant material, bacteria, apparently protozoa. I'm quite sure they're not selective at all. Yeah? And they simply will eat it and digest it. Yeah? And grow. Yeah. So where did you get your small glass containers? Is this a small petri dish? Yes, the small glass containers, those here. These are basically just small petri dishes, and you can order glass petri dishes, and you can order pretty much any size that you want. I also have larger petri dishes. Yeah. Over here. So for example, for larger specimens, you can get them over Amazon, and so basically, I always, when I use my stereo microscope, I always put the specimen on the petri dish, because it's simply much easier to move it around, right? Yeah, so that's the reason. So I highly recommend those. Yeah, there are also plastic ones available, but I like to use the glass ones because I just wash them out. Yeah, but it's as so often a question of personal preference. Yeah? So. Mosquitoes are lovely, really. Yeah. So there's some communication going on here from Tallahassee in Florida. Some hello. I recently made my own dark field filter with the help of your video. Okay, thank you. Thank you for the nice feedback. I recommend also that if you are successful already with dark field, try experimenting, also giving it the central disc, not a black disc, but some color to add a little bit of color it. Yeah. Check and correct the condenser. That's correct. Make sure that with the problem with the HDMI camera, make sure that the condenser is all the way to the top. Yeah, number one, and number two, make sure that the filter holder is all the way in. Yeah, so that you're not looking at not that the frame of the filter holder is kind of blocking some of the light. Yeah. Do you want to see the one that's in the dormant stage? Yeah, I can try to do that. And now I don't know if it's actually not already hatched or not. And it's pretty big, so I will do that as well. So what I'm going to do is I'm just going to now use the other. I'm just going to use now the other slide over here, the one with the concave. Yeah, and let's have a look at this one. So I think that should be a pupa. Okay. See, that's a little bit difficult to. I know those pupa are a little bit asymmetric. Yeah. Up. Oh, you didn't see that, okay, because it was covered. Yeah, so that's basically what I've got here. I'm just going to remove again some of the, and I'm not even aware if it's still alive or not, or anything. I'm just going to, let's just have a look at this. And usually it's so dark and large, so probably we're not able to see a lot, okay. So that's a little bit the thing, because you need a lot of light. But you know what? I'm going to try it first with a stereo microscope. let's try this first with a stereo microscope and let's see what this actually is. Pupa normally look like this, but I'm not quite sure whether this one is one. This looks a little bit almost like it's already hatched. I don't know. Yeah. Because I know that those the pupa of mosquito, they have these long extensions. This is honestly, could this be one? I don't know. You see the compound eyes here. I think this almost looks like maybe the leftover skin of a mosquito that hatched. Yeah, I think those round things, bulges that you see on the outside, look a little bit like those of the things of the compound eyes. It could be maybe an empty one, one after it hatched. Yeah. So, but not sure about this. Yeah. So yeah, so that's the thing. But you know what? Why not try it now also on the other microscope, just because for the fun of it. Oh yeah, where's this one already disappeared again? So this again in oh. Yeah, this looks pretty empty, I think. It's very thick. Yeah. So again, dark field. So let's have a look here at this one. Oh yeah, what's this? I have to find now the. I have to find. Ah, it's here on the side. Okay. Yeah. So what I'm going to do now is just to show you, for the sake of demonstration, I'm going to put now a cover glass on top of this one here. So I need to show you the desk view again. So it is here. Yeah, see, it's here now on the side. I have to somehow get it back to the. Or you know what? Maybe I can just add another one as well. Maybe I'm lucky enough to catch another smaller one. Yeah, there's a smaller one here. As a matter of fact, two smaller ones. Okay. So they're all in here now. Okay. Are there several of them now? Okay, cool. What I do now is I will now add a third cover glass on top. And this cover glass is a little bit bigger than the others. Yeah, so see. And one of the things that always kind of disturbs me a little bit, or not disturbs, but I cannot fully understand. Look at this. You just hold it against that's a fresh pack. Look how cloudy it is, right? Yeah, so there's always this, I don't know, grease or dirt or whatever on here. It's not that it makes a huge difference, but I don't get it. Why they can't just make totally clean, yeah, cover glasses? I don't get it. Yeah. So usually what I do is I dry wipe those, yeah, and just to remove the worst grease. And let's try it. Okay, let's try to put this over here, hoping, yeah. There's lots of water. You'll see sometimes this one over here is small enough that it's still able to move around a little bit. I don't quite. It didn't quite. Yeah, you see over here on the side here, it was not really. It's diagonal. It's resting on the slide over here, on the cover glass over here, but not on here. I should have moved it closer together. Not a big problem, but there is, in my view, in my opinion, still too much water here. So what you can do, there are two possibilities. I'll just use. You know what? I'll use this here. You can use, of course, tissue paper, or you just use a pipette to remove any excess water. Any excess water. Okay. Yeah, you see, this cover glass over here is it's not resting on this one over here. Yeah, so. Could be that some of those larvae over here are crushed a little bit more than, yeah. So, but let's have a look. The scope view again. Yeah, it's still dark field. Let's just go back to bright field again, just for right now. Let's see what we're able to find here. Let's go a little bit down with the light intensity, a little too bright. So here we go. Okay, here that's one. Let's see if I'm able to find others as well. That's one. Here's another one. Yeah, and another one over there. I don't know. Only two? I thought there were more. Huh. Okay, apparently only. Ah, this was not one. This was just a leg of an insect. That's why I thought it was one. So I think I have two of them here. Yeah. And one of the fun things here is to see that, is that the head, because it's so much larger, is not able to move, but the rest of the body is able to wiggle around. Yeah. So it's kind of like. So let's now, because we have a cover glass, we are able to go up with the magnification quite a bit more. Let's focus. This is how it looks like. I think it's kind of cool looking. I don't know. Yeah. So, and I personally consider is always kind of fascinating. I mean, these larvae look so different. I mean, it's of course the case with many insects. They look so different from the adult insect. Yeah. That metamorphosis is really fascinating. So this back part here, this is the part that they're putting into the air. So very often those mosquito larvae, you can actually see them dangling with this back part sticking out into the air and the heads downwards, right? Because that's kind of the breathing organ here in the back. And then it will go into, when it's of sufficiently large size, it will go into a pupa stage, and then after some time, I don't know how long the time takes, several days. I know with flies, it takes around 21 days. Yeah, they hatch. Yeah. So I'll quickly check again some of the questions that you might have. Yep. How does a larva in the pupa transform in an adult insect? Does the adult grow in the pupa eating the larva? In other words, what's going on in the pupa? Well, that's the cool thing. No, it doesn't eat. The thing is it actually, how shall I put it? It actually changes. It will grow wings, it will grow legs. Yeah. So that's basically what happens. It will actually transform itself. Yeah. It's like a butterfly. Yeah. And then basically there is this not shell, but the skin on the outside, and then it basically hatches out, and out comes the adult insect. That's actually one of the most notable one is what happens with butterflies. Yeah, because it's such a huge transformation. Yeah? So that's why metamorphosis, as it's called, it's a change from a larva stage to over a pupa to the adult. Yeah, that's why metamorphosis is so fascinating, and that's why butterflies sometimes also has a strong cultural tradition that if you need some kind of a metaphor for transformation, for change, usually the butterfly or other insects are used as an example for this, because the change in shape is so extremely dramatic. There are other insects out there where the change in shape is not so dramatic. For example, grasshoppers belong to that. Yeah, the larva stage of a grasshopper still looks kind of like a grasshopper, just in a smaller version. But there are insects like butterflies, or in this case, for example, flies, and also those mosquitoes are also related to the flies. Obviously, they have a complete transformation, and it's very different. So it is not like this that the adult kind of eats up the larva or so, but the larva actually itself changes. Yeah, it will grow wings, it will grow legs. Yeah, it's a complete change in body morphology. Yeah, so this is the fascinating thing. How does this work? Of course, this is intensively studied. This is if you want to have a biological term, it's a question of the so-called gene expression, as it's called. I have my molecular biological background, so I cannot avoid using those terms. Basically, it means that certain regions of the DNA of these organisms are switched on and other regions are switched off. Yeah, and this has been intensively studied also in fruit flies, the development of the fruit fly, Drosophila, for example. Yeah, and because it also undergoes this metamorphosis. And how is this controlled on a genetic level? How can you actually tell certain cells that they should form now a wing or legs or other body parts, right? And this basically means that chemically, certain parts of the DNA are switched on that are responsible for that. Other parts are switched off. And this whole field of study of how can you switch on and switch off the DNA of the different cells is referred to as a gene expression, the study of gene expression. It's a huge field of research. in molecular biology. Yeah? And why is this important? Well, it's has an understanding. This has also direct importance for understanding how we humans work. I mean, we humans ourselves started off as one egg cell, a fertilized egg cell. Then we were a ball of cells, yeah, an embryo, and all of the cells were the same at the beginning, right? They were all the same cells, but then later on, some cells developed into the head, the other ones, yeah, the other body organs. And how does this work? Yeah, how can the cells now start to become different from each other? Right? So if you want to see this change happening, as interesting as it is in studying it in those insects, like in the mosquito or butterflies, we also have those radical changes during our own development. Only it's not as visible because it happens when they're during pregnancy, so we cannot see that. Yeah? So it's not as evident, but those big changes also happen in our body during embryonic development. Of course, differently, but the principles that cells change, that's the same, that they differentiate. Yeah? And for this reason, studying how or understanding how does it work in insects, if we understand that, then we can also understand a little bit better of how our own body develops, right? So I know I'm going off into a little bit of biology. I hope you don't mind. Yeah, but that's also one of the things that when I look at how do you say, and at those insects here, like mosquitoes, and the development. I don't only see a mosquito larva over here, but I'm also seeing, and that's a little bit how I'm conditioned, certain similarities with other organisms, including us humans as well, right? Because on a very, very cellular level, if you go very deep down into the cells and into development, there are a lot of similarities. So the deeper you go, the more similar things become, yeah, on a biochemical level. Yeah? And so even though we're so different on the outside. Yeah? So it's, yeah, I know I'm deviating off a little bit, yeah? Here. Is the bright aura around its head an air bubble? I don't know. This question was posed maybe a couple of minutes ago, so it might have already changed. Over here, I do not see any air bubbles. Air bubbles are very easily visible because they are not only quite round, but they're very disturbing. So basically, they change the refraction around quite a bit. Yeah, I cannot see, but you know what? I just go to the side. I don't know. I don't see any air bubbles here now. Yeah? But if I do see them, then yeah. By the way, in the video that I made, the other one about those mosquitoes, there is actually on the back side a tiny little air bubble. Huh? So yeah. So doesn't the mom feed them blood? Actually, as a matter of fact, no, because that's the okay. Also interesting question. Is this the reproduction of those? There are essentially in nature a variety of strategies, reproductive strategies. And there is one extreme case where you've got parents who have few offspring, few babies, few children. Humans, for example, in extreme case. Yeah, so essentially in a family, just maybe 1, 2, 3 children, 5 children maybe, and that's pretty much it, right? So throughout a whole lifetime, but the parents invest a lot of energy into those children. And on the other extreme, you have cases like, for example, insects, where there are many larvae formed, many eggs formed, many larvae, many babies, many offspring, but the parents invest practically no energy in them. So basically, only few of them will survive. Many of those mosquito larvae will probably be eaten by, I don't know, other animals, frogs, maybe birds, I don't know, other insects maybe. Yeah? So this is a fish, definitely fish. Yeah, some people like to grow those mosquito larvae at home because they have an aquarium. Yeah? So these are basically two strategies here. In nature, you have basically animals that invest a lot in their offspring, right? Humans being, in extreme case, few babies, but the children stay attached to the parents for a long time. And then on the other extreme, you have those animals where they have many offspring, thousands, hundreds of thousands, millions maybe. Yeah, and but very little energy is invested in their survival, and many of them are basically eaten. Yeah, most of them are eaten. and a few of them survive. Yeah? So it's a statistical question. Yeah. So that's shortly the question if the mother insect doesn't feed them blood, and say nope. Okay. Since tardigrades are not microorganisms, what are they? Microanimals? Yes, as a matter of fact. This is not my definition, but this is actually what I also read. Is specific. Tardigrades are considered to be microanimals because they are real animals. This means they're made of around 1,000 cells. And the cells are different from each other, differentiated, yeah. So specialized, there's cell specialization. And in microorganisms, essentially, when they divide, then each cell is able to survive on its own, so to say. Yeah. But in the case of microanimals, you have the cells that are kind of make up one organism. And there is an interdependency, so you cannot simply take one cell of a tardigrade and it's able to survive. I'm not able to take one of my skin cells, for example, and it's not able. It won't be able to survive. Yeah, so that's kind of the maybe one of those criteria. Microorganisms are single-celled, and the individual cells are able to survive on their own. Yeah? So. I'm going to go down again some of those questions, and then actually I'm talking so much. Actually, I also want to show you an adult mosquito, and I would like to mount one. Yeah, I'm just skimming through. Maybe there's a look inside a microscope slide cover glass making factory. Yep. Yeah, that would be interesting to look at how do they actually make microscope slides and cover glasses. Yeah. So I'm just. Does this larva have a nervous system? Definitely, yes. Yes, of course. Those larvae have a nervous system. Yeah? They have a coordinated body movement. Look, they have eyes, they have antenna. All of those hair on the side that you see, I'm quite sure, are sensory hair. So they have a nervous system, and yes, of course, the adult mosquito also has a nervous system, because they have to be able to sense, they have to smell where is the food, where is the blood, right? And so they are, as a matter of fact, they have very, very sensitive sense organs. Other insects, also for example for mating, one of the reasons why those mosquitoes have those gigantic antennae is not only that they're able to smell the blood, so to say, or the people, or the sweat of the people, right? Sometimes they smell that, and then they're able to find the food, but sometimes those antennae are also used to find mating partners over so-called pheromones or chemicals that are released so that insects are able to find each other for mating. And they're extremely sensitive, and for that you need a nervous system. And yes, they have a brain as well, but a very simple brain. Yeah, but obviously. So I know now, of course, this begs the question: if they have a nervous system, what are they able to subjectively perceive? Are they able to perceive something like pain or things like this? And that is, of course, something that we probably don't know, because we are not able to look into those insects. So concerning subjective perception is, of course, something that we're not able to know. Yeah? But the answer, or the question is do they have a nervous system, is a very clear yes. It's a very clear yes. Yeah? They're invertebrates, but they have a nervous system. Yeah? I bought the microscope question again, the SW380T, having problems getting dark field to work with a filter. I found that the condenser is way off center. I have got it closer but cannot fully center it. What can I do? Of this SW380T, the condenser is fixed with 3 screws. And those 3 screws, you just loosen them. You do not remove them completely. And if you loosen them, you can recenter the condenser. It is made in such a way that the holes are bigger than necessary so that you're able to move the condenser around. It has been actually designed in such a way that you're able to center it manually. Okay, so that should work. You have to find this hexagonal wrench type of tool, and you just loosen the 3 screws on the bottom of which holds the condenser in place, and then you're able to move it around, and then you're able to tighten it. Yeah, and so it's fairly easy, and you can basically know when it's centered by simply closing the condenser completely, and then you should be able to center it. Okay, but that can be done. How do I know? Because I tried it. Okay, and it's because it's such a common question. I have to tell you, that's a little bit of a, I would say maybe a sloppy assembly, that they have not pre-centered it. Or sometimes it's a question of luck whether it's centered or not. But generally, it shouldn't make a huge difference because sometimes if you do not put in the filter holder all the way anyway, the filter is also off center anyway. So it's not a big thing. Yeah. Yeah. So I'm going down here. And then since they have a brain, do they keep the brain after the metamorphosis? Yes. Yeah. Well, of course, the brain also undergoes changes. Of course, must undergo changes because afterwards they have wings. So you need to coordinate the wings, the flying patterns. They need to be able to sense the blood. So does the brain undergo metamorphosis? Of course, it must do that. But does it have a simple brain? Also, yes. Yeah, I mean, sometimes you don't call it really like a brain. Sometimes you call it some kind of like, it's called ganglia. Ganglia is an accumulation of nerve cells that you have, which is kind of like a precursor of a real brain. Yeah, but what we have here is what, I'm going to use biological terms again. It's what you call cephalization, it's called. Yeah, so for example, if you look at other animals like starfish, for example, those five with the 5 arms, right? They don't have a real brain. You cannot say where is the head part and where is the tail part or the front or back part. You cannot say that in certain animals. Or jellyfish, for example, same thing, right? You don't. Yeah, you have certain. It's highly decentralized, the nervous system. But there are certain animals like worms, for example, or those insects like the larvae over here. You can clearly see there is a head here, right? Yeah, and somewhere in here, and I'm going to turn on now the arrow. Where's the arrow? Oh, gee, yeah, somewhere in here. What you see in here? Just a second. How can I? Here's the arrow, right? You can see, of course, the pumping. Maybe, I don't know, this could be the digestive system. Maybe it could be they have a simple, you don't call it a heart, but actually certain blood vessels that are actually able, well, they have an open circulatory system, right? Yeah, as well, and it can be that over here somewhere there must be certain, how do you call this, nerve cells running through here, right? Because look, this is a coordinated movement. What we have here, this must be controlled. Yeah. So, but also very clear, the brain is definitely there. Yeah. Yeah. Interesting. Oh, I love philosophy here. I love philosophy. Here's the question here. No, we don't care about pain. We want to know if they have a favorite color. It's interesting that everyone wants to know about pain. I can't tell if it's to project morals or if it's darker. Well, I will tell you the following. As a teacher, we're catching insects. We have an insect project going on every year. So there's an entomologist, an insect specialist helping us, and he helps us basically to catch insects, to release those that are protected. That's also important. So for example, we do not catch bees, and then we have to kill them so that we're able to observe them under the microscope, right? And indeed, there are people out there, students out there who don't want to do that. And they say, well, actually, is it the morals, is it okay to kill animals for this? And I say, come on, do you eat meat? Yeah, seriously, yeah. That's one thing. But I think some people are a little bit upset about this because they're in, I don't know, maybe because they just see it and then they kill it, and maybe they don't like doing that, right? But I personally have to tell you the following, and I mentioned this already before. I actually do have ethical problems when people are worried too much ethically about microorganisms. I mean, we have to protect the environment, of course, but honestly, because of environmental destruction, there's so many animals displaced and killed and extinct and so on. Why is nobody caring about that? You see, you worried about mosquitoes, and other insects and so on, and whether they feel pain. But what about the real issues? So this is something that I tell my students. Yeah, if you want to be moral or ethical, then be moral and ethical about the important things. So in that sense, I have to agree with you, with Josh. That essentially sometimes I think it's a little bit overdone. Yeah. So can anyone cephalization? It's spelled with a C. Yeah, with a C. Cephalon is the head. Yeah, encephalization is the formation of a head, so to say. Right? Or simply the accumulation of nerve cells. Yeah, yeah. So yeah, that's what it's called. So cephalization is not spelled with an S, but with a C. Yeah. Yes. Oh, thank you, Josh. Okay, again. Yeah. Yes, thank you. I didn't see this comment. Yeah? I didn't ask for nervous system because of the pain, but for my next question, if they have a brain and keep it after metamorphosis. Yes, no, the point is with the pain aspect, I didn't want to project it into you, but I'm just saying that this pain question is one of the most common questions that I'm receiving from students. Yeah. So usually if I say they have a brain, they have a nervous system, the next question that usually 90% of the cases appears is, well, do they actually feel pain when we kill them? Right, and that's so it's yeah, that's simply where I kind of picked up. Yeah, so yeah, so this is the thing here. Yeah, so I'll be honest with you. I don't feel sorry about mosquitoes because honestly, those mosquitoes they transmit at least in the tropical regions so many diseases. So let's feel sorry about the animals or humans that die, yeah, or become ill. Yeah. So I'm getting lost again in sidetracking. What I'm gonna do? Oh, there was something floating around here. This is kind of fun. Maybe some protozoan or so. I don't know where it is. Occasionally you see some objects swimming around. So what I'm gonna do now is I'm going to yeah, I'm gonna put them back into the water, not because I feel sorry for them, but because I want to keep them in case I want to do some more microscopy with them. So, but I'm not gonna, yeah, I don't know how am I gonna do this. So I'm going to carefully bloop, and I'm going to dip it in here. Yeah, here we go. There's one left over here. Let's put this away. And let's just rinse it down. Yeah. I think they'll do. Okay. So maybe after a day or two, some more mosquitoes will come out here. I'll put this away. To prevent the evaporation of the water, I put a thing on here. And then I now want to show you, and yeah, where is this thing? Here it is. Where is the stereo? Here we go. Here, this guy over here. Where is it? Okay. This doesn't. What is this? Whoops! It's embarrassing. What? This doesn't look like. Oops! I caught this was flying around. This does not look like a. Look at the mouth parts. This is not a biting mosquito. Oops. Yeah, you see that's how bad my eyes become. Mosquitoes have this long, how do you say, proboscis. Okay. This one doesn't look like it. Look what I've, luckily I do have a reserve. This one is dead already. Because, and look, that's how I think a mosquito should look like. Yeah, so, okay, there were so many, I caught this just before the live stream, right? And it was flying around, I quickly caught it, I finally got one. And then now looking at it, I discovered it doesn't look like one. Okay, it doesn't matter, we can still have a look, but that's the spear one. And let's look at this thing here. Okay, let's focus. Do you see this long thing that's kind of extending here? Where is this arrow? That's the, yeah. It's for sucking the blood, right? Yeah. And it's a little bit unfortunate that yeah. So that's what I call yeah. And another thing that I would like to mention here is the following: is how many pairs of wings do mosquitoes have? Mosquitoes, just like the flies, belong to the so-called dipteras. Dipteras are those insects that have only one pair of true wings. Okay, so for example, if you look at bees and honeybees and wasps and so on, even butterflies, they have two pairs of wings. Yeah, yeah. So, but those, yeah, mosquitoes only have one pair of wings, and this is the, how do you call this? The proboscis, which is kind of, yeah, for sucking the blood. And what you have up here, these are the antenna, this here and this here, right? And the other thing that I caught, and I said, wow, so evidently this is next to the water sample that I had. Evidently, there are lots of other insects as well that kind of, yeah, reproduce in water. Apparently, and this one wasn't, I don't know which one this is, but just from the mouth parts, yeah, here it is. Yeah, I don't even know. Yeah, also, I don't know, it also seems to have, I think, one pair of wings. Yeah, but it doesn't matter. Why did I say true wing? Okay. Because the second pair of wings are so-called halteres. So this is a little bit, I need to, and I don't know if I can see it here. Flies, if you put a fly under the microscope, you're going to see that essentially there's a wing, and the second pair of wing is so reduced that these are just like club-shaped structures which are kind of vibrating to keep balance, but these are not really wings as such. Yeah, so that's why I said true wings, because from a developmental aspect, you could call it these halteres, as they're called, are actually like the second pair of wings, but actually they're not wings because they're not used for flying, but only for balance. Yeah, so that's essentially the reason why I said that. Yeah. So that is, yeah, that's why I said kind of true wings. Well, what I would like to do now, therefore, is I want to show you, what time is it? Gee, I'm spending more time on this. I actually also wanted to show you the cherry and the leaf I can do very quickly, okay? But I'll just want to show you a little bit how I prepare those things here. And what I need is the following. I just want to put a wing now under the microscope. And I'm looking for something. Where is this? Here it is. I found it. Okay. And now the thing is I want to cut. Oh, I dropped something. Oh, nothing. Okay. So what I would like to do is I would like to put this back here, and I would like to separate. I would like to separate now. A wing here. And the problem with that is that my scissors are too big. I just want to show you. These are the finest scissors that I have. You know, they're pretty pointed. But now compare this. Now if I put it here under the microscope, how big they are compared to the to the wings, right? Yeah. So if I want to cut off the wings here, it's I don't have a lot of control. So this just shows you a little bit the size difference. So I found an interesting new way. Interesting, it's not interesting, but yeah, a new way. Because of what I have here, which I bought some years ago, these are insect pins. And they're much thinner and smaller than regular pins. And actually this is I can use those to separate the wings from the, from the mosquito. Okay, so let's try this. Okay, so you see it over here. I have to, okay. You don't mind that I'm going a little bit off screen. Okay, so what I have to do is, you see? Okay, so it's kind of not very tight. So what I have to do first is I'm going to try to pin it. If I'm able to do that. Let me quick, see, I have to I have to hold it down somehow. This is very difficult to do that here now directly. But I might be able to maybe hold it down with one needle and then maybe try to separate it with the other. This is, at least one of the legs went off. Yeah, this is a question of patience. You know what, I'm going to do this the very brutal way now because I don't mind destroying the body. Look what I'm doing. I'm just going to do it like this. I'm going to hold it. And I'm going to try to separate. It went off. Beautiful. See? Where is it? Here it is. Here's the wing. Okay? So that's what I wanted. It's already so dry that they break off quite easily. And now the question is now how am I going to get this? Let me put the needles away, otherwise they're so sharp that I might hurt myself. So how am I going to get this now on a microscope slide? You take a microscope slide. Okay, like this. And where is the desk view? Here is the wing. Okay, here is the microscope slide, and I'm using my favorite mounting medium here. And I add a very small amount of mounting medium. It's way too much. So a very small amount of mounting medium. And now I somehow have to get the wing across. How am I going to do that? You need a toothpick, which I don't have. Great. What I'm going to use instead, I'm just going to use I'm going to improvise again. I'm going to use one of those tips here of my disposable tips. And what I do is how do I get it across? I dip it into the mounting medium. I transfer the wing in here. And you know what? Why not also have a look at the leg? Okay. So, and then I usually I trash it. And then I usually add a little bit of mounting medium also on top. Okay, to make sure that, yeah. And next thing, and that's usually the risky thing, is when I add a cover glass, usually what happens is that both wing and leg are pushed all the way to the side. So this is a little bit a question of luck as well. Yeah. So I want to try to place it directly on the center. Okay. And let capillary action do the rest. See how it kind of, okay, it stays pretty much in the center. And you notice that no mounting medium appears beneath. That's good. And this basically means that even before it has dried out, yeah, I'm courageous enough to put it under my microscope because you do not want any mounting medium to get on your objective. So let's have a look how this looks like. So again, let's like this. Ah, yeah, here is the leg. Yeah, that's here. Now you see some of those round things. These are the bubbles, right? And the long leg. Here it is. Okay, tiny little claws at the end, and the other side of the leg. You see, that's also one of those things. Do you see those white regions here? This can actually be used also for identifying the mosquito. And it could be, the mosquito's genus could be Aedes, I think, as it's called. But there are several mosquitoes that have those white patterns on the legs. But I'm not an insect specialist or a mosquito specialist, right? Yeah. But this kind of shows here a little bit how the leg looks like. And again, this is very different from the larva. Okay. And where is the wing? The wing is over here somewhere. Okay. Yeah, so again, those round structures are bubbles. So it kind of shows that the mount is not very perfect. But I think you get the idea. I think that's the point is that you kind of see how it looks like. And those scales, those tiny little scales that you see, which also come off, these can indeed be found fairly often also in dust samples. Then they're not necessarily always from mosquitoes, but also butterflies have one that kind of looks similar. Yeah. So if you ever see those structures, then you know that these are some insect scales. Yeah. Yeah. So that is so let's have a look again. Some of the questions here. There are lots of questions. I hope I'm not missing anything. Okay, I'm going to simply skip over most of them unless, okay. I've seen one of your videos where you recorded your blood for half an hour and then sped it up to see the white blood cells moving. How did you prevent it from drying? That's an interesting one. It is like this, that I prevented it from drying by using a fairly large cover glass, and the blood will dry on the edges, okay, of the cover glass. There's no question about that. But when it dries on the edges, then the central part remains liquid, because when it's dry on the edges, then the central part is not able to evaporate. So that's kind of all I did. Yeah. So I did nothing essentially. Yeah. So that is So let me have a look here. Yeah, and yeah, exactly. With LED microscopes, which produce relatively cool light, it also doesn't dry quite as quickly. Yeah. Uh, so, so that was the whole question with the wings. I go down again. Could you increase the resolution of the microscope by using blue light or shorter wavelengths? Yes, but the question is whether it really makes a huge difference for all practical purposes. I know that some people are actually using blue light because they're able to make some of the structures more visible, but I don't think that this will be so huge. Yeah, I don't think that it will make a huge difference. Yeah? Are mounting mediums expensive? If yes, can you use any cheaper type of oil, cheap oil? Short answer, yes, mounting media can be very expensive. That's not the problem. If you've got one bottle, then you can use it for a long time. The problem is that many companies won't sell them to you because you're not a lab. And therefore, I don't know, that this, I've said, that this is, I'm not sponsored, but this is actually a glue, Elmer's glue, and that is PVA. And this is actually a mounting medium that I recommend. It's just children's glue. It's completely clear, not the white version, but the clear version. You dilute it down with a little bit of water, and this also works. That's not the one that I've used now, okay? I've used now U-Pearl mounting medium, which is always used for insects, but this one also works. Yeah, so, and this is very cheap. And I would probably, before trying to get any expensive ones, I would actually get this here, a bottle of this. Huh? So, and then dilute it down maybe with about 20% of water you add, you mix it, and then you've got a mounting medium. It works surprisingly well. Yeah? A bad result with Elmer's glue with stems. It's because it is not suitable for wet mounting. Well, actually, Elmer's glue is water-based. Yeah, so it should not. Yeah, it should be compatible with a wet mount. But it could be that if you actually had too much water in the specimen, then it could indeed be that it did not mix properly with the glue. So that's another thing here. How can I slow down the movement of a single-celled organisms? They are very fast. Yes, you can do the following. You use very little water, very little liquid, and then the microorganisms, paramecia, are actually squashed between the cover glass and the microscope slide. Yeah. So, and this is actually slows them down quite a bit. There are also substances like methylcellulose or something called Proto-Slow. It's a commercial product that you can add, and it's very sticky and viscous, and this slows it also down. Yeah. You can put samples in the fridge. Yeah, some of them will slow down because generally, if it's colder, then the metabolism slows down as well, and therefore some organisms actually move slower. Yeah. So what I'm going to do now is, what time is it? Okay, it's already over 1 hour. I spent more time on this than I actually intended. But what I would quickly like to show you, and I'm going to keep that short, is, video will follow. I would like to show you how to quickly, just for your fun a little bit, how to make the desk view. So complete change in topic. So let's put the mosquitoes away. How to make the surface of a leaf visible. Again, I know that some of you might already know this because I've done this already in the past, but maybe some of you don't know it. I've applied a little bit of this glue here on the bottom side of a leaf. The bottom side is important, right? And when you let it dry completely, and if the bottom side of the leaf is sufficiently smooth, then you can, after a day or so, you can carefully try to peel off the glue. You see how easily it goes off? You turn it around, and this is the bottom side now, which was in contact with the leaf, and you put it as it is on a microscope slide. And you hope that it's kind of reasonably flat. And you put that under the microscope. Okay? So I'm just telling you because some folks, maybe you want to play around a little bit, experiment around a little bit. And I've tried a variety of different glues, and all of them worked. This one is now, again, the Elmer's glue. I tried some white glue, wood glue works. I tried, I don't know, all sorts of nail polish. I think might also work a little bit. Yeah? But in any case, now let's have a look at this here, okay? And I'm going to show you some. So again, here what we're looking is just the surface impression of the leaf on the glue. Yeah, so we're actually able. We're just looking at the shape of the glue. Okay. So and let's do that first. And this is what we see. Okay, a whole bunch of dots. Okay, and so I'm going to explain you a few things here first, because where's the arrow? The arrow is here. This is, for example, these are the veins where water is carried. So this is the surface of the leaf. Okay, so you see that. But what we do is we zoom in now more. No cover glass or anything, right? Okay, and what we actually see is now the individual cells. Yeah? And what I want you to do is I want you to have a closer look at those oval round structures that we see here. There are quite a few of them. Yeah, these are basically somebody says topography. That's correct. Yeah, so this is now the 40 times objective, and I'm not going to go any further. Yeah, but basically what these are the so-called stomates or stomata, and these are openings on the bottom side of the leaf. And essentially what they do is they're able to open and close to let moisture out and especially air in. So air with carbon dioxide gas goes into the leaf through these openings here. And photosynthesis is able to take place, and oxygen, which the plant produces, goes out. So this is kind of the topography indeed, and left and right of these openings, there are so-called guard cells that are able to open and close the central hole here. So this is just a very easy thing to do, and it works surprisingly well. And so also for educational reasons, I mean, I'm a teacher and so on. And when we talk about photosynthesis and leaf anatomy and leaf structure, and that's actually one of the things that I like to also show them, because you learn about this in school, those stomates and the openings and the gases that go in and out of a leaf. But actually it's really easy to make it visible, because trying to cut those leaves, let me move the microphone a little closer, trying to section them and microtome them is pretty difficult. I tried to do that, and but actually I'm getting much better and more interesting results, also maybe even prettier results using the glue method here. Yeah? So just wanted to show this to you a little bit, and to encourage you to also try this out. Yeah. So let me quickly go through this here. Yes, there was water on my specimen, so you suggest me to dry it out. No, yes, okay. So this goes back again to the mounting medium question. Don't dry it out completely, but make sure that there is. But what you can do is you can use a little bit of tissue paper to remove all of the excess water. Because if there is indeed a droplet of water around, or if it's really wet, then the glue cannot reach it. Yeah, and if you dry it out first, you can try that. It depends on the specimen, but if you completely dry it out, then it might actually shrink and it might also deform. Yeah, so it is a little bit. It depends a lot on the specimen, how sensitive it is to drying. Some specimens really don't care if they're dry or not, and others they shrink quite a bit. But what you should try to avoid is if it's actually really wet in the sense that there is some kind of a visible droplet of water around it. Otherwise, I would actually use some tissue paper and remove that, or dry it for a couple of minutes, but not completely. Okay, it's experimentation. Yeah. Are the veins hollow structures that reach and are hollow all the way down to the roots? In other words, what you're asking me, I reformulate, is what is the anatomy of the so-called the xylem tissue? Xylem are the tubes that carry water up a plant, and indeed these are hollow cells. Okay, these are dead cells that are run in long tubes all the way to the top. Yeah, that's actually correct. Yes. How would you make a permanent liquid blood slide? What you do is you cannot make it permanent as long as it remains liquid. You have to dry it, you have to air dry it, and then you add some mounting medium on top and then a cover glass. But as soon as water is present, the slides generally are not permanent, because as long as water is present, bacteria can decompose the specimen. So in order to preserve specimens, either on a microscope slide or generally it has to be dry. I always use the example, why is it possible that potato chips, why don't they turn bad? The reason is because they're completely dry, there is no water present, and therefore bacteria cannot grow. Yeah, so that's kind of the thing. If you want to preserve specimens, you have to make sure that they're free of water. And if you add now a water-based mounting medium like the glue here that I showed you before, it will dry. And when it's dry, then also bacteria cannot break it down anymore. Yeah, so that's a little bit the thing. Yeah. So can I put U-Prol on it? I could, but the problem is the specimen is too thick. Okay, it's too thick, and therefore it's going to, there's going to be a lot of this U-Prol or glue around it. Okay, but I might try this first, and then maybe next time I'm going to show you. So I'm going to move on now, because I want to now, and it's something I did not try yet, and I have to try it now, because I have to throw it away later on, and that's basically the cherries. Okay, so let me put the needles away, the dangerous needles here. Okay, that I showed you. I have to make sure that they're properly aligned first. Just a second. Okay. Because if they're okay, yes. So here we go. I put this away, and here these are the cherries. There are lots of fruit flies already also on here. What do? Is they lay eggs down here? Yeah. And the larvae come out, they go, oh yeah, you get the idea. This is beautiful. Not healthy. Why not? Number one, the spores, if you inhale them, not good. Number two, fungi, like many other microorganisms, do produce certain toxins or poisons. That means that even if you were to boil these things, it could still be bad for your health, because those poisons might be heat resistant and you don't want to eat it anyway, right? So what I would like to do now is the following. I'm going to put it under my stereo microscope first to see a little bit how this looks like, okay? So this means stereo view. This plate goes all the way down here, and I have to move up the focus. And we are already able to see that what I wanted to show you. Okay, that's the nice. Let's zoom in a little bit here. There where the contrast is nice. So let me have a look here. And so you see. Okay, so just a few nice things here you see over here. Do you see this tiny ring here in the reflection? That is the ring light of my lamp. And those bulbs, those bubbles that you see here, they seem to be the so-called the fruiting bodies, or the spore-forming structures. So what happens is that inside the cherry, you have the fungus growing, and then they will grow essentially those structures, and in them there should actually be the spores. Okay, so this is a little bit the thing here. I remember just today in the morning those cherries looked much better. You have got exponential growth here. Okay. But essentially under the stereo microscope we were able to see already quite a bit, here, so that's pretty dramatic, Not this stuff ain't healthy, right? So this is the reason why you have to be really careful. I mean, I don't know which species they are. What I heard is, when I still studied microbiology a long time ago, is that of course, just like with many other microorganisms, the identification of fungi and so on, just using a microscope is very, very difficult because there's simply so many of them. So maybe you're just able to somehow get the direction of yeah, but not all the way down to the species. You'd have to do probably a chemical or DNA analysis to actually determine what it is exactly. Yeah, but it doesn't matter. What I'm going to do is I'm going to take a few of these things and I'm going to also put it under my compound microscope. Yeah, my goodness, the cherries, the fungi that have been viewing under the epifluorescent to see the fungi, yeah, on live root samples. Interesting fluff. Okay. Ah, yeah, Matt Powers. He is. I've also watched his YouTube channel. Yeah. Yes, it would be kind of nice to film a time lapse. Indeed, this is also something that I've been thinking about. It would be kind of interesting to not only film a time lapse of those cherries decomposing, but actually also under the microscope. Yeah, because yeah, this should be quite nice. Yeah. So you won't show a sample of the 100,000-year-old oak tree, only the impression. So okay, I'll show you something. Okay, the oak tree. Look what I got here. This here is the leaf of the oak tree, which I put into alcohol to take away the pigment, the chlorophyll. And I'm trying to still work on a method to get proper cross sections of this leaf here. Okay, I know that that's kind of the thing, but I've tried this and getting very thin, nice sections. I was not satisfied with the results. Okay, so that's why I've only done the glue impression here. Okay, so what I'm going to do now is, this is, yeah, almost like cotton candy. So what I'm going to do now is I'm going to take a sample here. You got to be careful. Look, with those tweezers, when you take this stuff, you have to later on I have to really clean it, because I want to get all of this stuff away, and this goes then down into the how do you call this? Not the trash, but into the container for compost. That's the thing. Huh? So we take a slide. Okay, take a slide here, and what I usually do is the following. I usually take a small sample. A droplet of water first. So what I have over here is okay, so let's add a little bit of water, just regular water. There was already something in here. Okay, and let's use the tweezers here. Okay, something like this. Oh yeah, you can see it because it was, yeah. And now I'm going to try to take carefully, without doing too much damage to the structure. I'm almost blind. Very difficult for me to see this, if I actually got something here or not. You don't really need a lot. Let me see. I think I do have, yeah, there is something on here already. Okay. Okay, so I have to now, yeah, clean the tweezers. Here is my bottle of alcohol. Okay, and I'm going to just disinfect them because I do not want to hurt myself, yeah, with this sharp. Look, do you actually see how much there is on here? Yeah. So let's clean it properly, and I remove this here. I close it again. And we add a cover glass. Okay, and yeah, it's maybe a little too much water, I don't know. But let's put it now under the microscope. Let's hope that I found enough of these things here. I don't know what we're able to see here. Oh yeah. Ah yeah, those dots here, air bubbles. And that's the stuff that we don't want to inhale. These are basically spores. Millions of them. Yeah? So that's the reason why we have to be a little careful of keeping those moldy vegetables and fruits and so away, because those spores will of course spread. Look at this. This could be actually one of those spore forming bodies, conidia. Those round thing, and sometimes they pop open, releasing the spores. Yeah. So that's kind of the thing. What I would like to try is, and that's kind of a little bit my goal, and I was not successful all the time, is to actually germinate them. But look at this one over here. Maybe. Do you see this guy over here? You see that there's a tiny little outgrowth? It's worth maybe following those a little bit. Maybe it will actually start to grow out and start to form a new hyphae. Hyphae are the cells of a fungus. Yeah? So this is actually something which I would also like to do in time lapse sometime. And I've seen this already under the microscope, where you could almost watch this. The growth was very, very rapid. Yeah. So these spores essentially they spread over the air, and when they fall on another fruit, on ground, or wherever which is suitable, then they will also start to form a new fungus. Yeah. So that's a little bit the thing here. Yeah. It could be as well that because I added now water to the whole thing, is sometimes what will happen is that those spores will start to absorb the water and start to change their shape because they start to swell. So sometimes their shape looks different, or might look different, depending on whether it's in water or whether they are dry. Yeah? Yeah. You think you saw a pollen grain somewhere? Could be. Well, pollen grains should be a little bit bigger, most of them. Yeah? But that's possible. That's quite well possible as well. So, yes. It's the question. There was a thank you over here. Okay. Yeah. So that's a little bit the thing. So where maybe we can find. I don't know where it was before. But usually this is something that you find on many fungi, except you might be a little bit surprised. I also made sometime a video ago. If you try to put now camembert fungus under the microscope, then you're going to see the individual cells, but not many spores, if any at all, because the camembert fungus is in such a way that it's already, it doesn't like to form spores anymore. So it's kind of a little bit overbred, as we say. Yeah. So some of the folks are actually a little bit concerned that it might be difficult to reproduce the camembert fungus in the future. Yeah. So okay. So this is actually a nice example to show you mostly spores, but few hyphae, which are the other cells. Yeah. And the reason is because those hyphae, the cells of the fungus, is actually growing into the cherry. Yeah. So this means even if you see something that's, if you see some food that's moldy on the surface, actually the fungus itself will grow fairly deep into the food. Yeah. And so simply cutting away the top or the moldy part is not going to work. First of all, because the poisons that are made, they will spread throughout the food, right? And that's something you don't want to eat in any case. And also because the cells of the fungus will also spread into the food and release the enzymes for digestion and so on. Yeah? What happens if alcohol meets the spores? Basically, it depends a little bit on the cells. It is like this, that spores generally are pretty resistant to drying, and however, if alcohol meets other cells, sometimes they will dissolve, right? And the cell membrane will dissolve, and they will pop open. But it could be, and I don't know, it could be that the spores here will not pop open if they're in contact with alcohol, because generally spores are quite strong. But you know what? We're going to try that out. And that's going to be the last thing today, because I'm already talking for, 1.5 hours. So I'm going to add a little bit of alcohol here. And what's going to happen now is when I add some alcohol, and I have to use the lower magnification here, because otherwise, I might get some liquid on the objective. And what's going to happen here now is when I add the alcohol, you're going to see that the alcohol and the water will mix, and there will be some kind of turbulence and movement, right? Just so that you know what to expect, and that is because the alcohol is kind of breaking the surface tension of the water. So I'm going to try to add now a little bit of alcohol. Let's go to the corner here. Okay, so that's the corner, right? The edge of the cover glass, right? And this is where I'm going to add a little bit of alcohol now. Okay, and you see those? Did you just see this swirling movement? It's because of the breaking of the surface tension, and you see actually sometimes the question is if the alcohol is also able to enter this is because of the changing refractive index of water and alcohol mixing. Yeah, but this is not where the cover glass is. Yeah, but maybe we're able to see a couple of spores and to check what's happening with them. Yeah. Very often the alcohol will actually remove water from the structure, so it could also be that the spores will start to shrink. And I've also seen some cells, when you add some alcohol to them, that the cells will actually pop open because the alcohol has dissolved the cell membrane. So it really depends quite a lot on the actual structure. Okay. See this, all of these things. That's the water and the alcohol mixing. But actually, if you just look at we can go up a little bit with the magnification now. Yeah. So just for your orientation, this here is outside of the cover glass, and this is still beneath the cover glass. So the alcohol is still mixing on the outside and slowly moving in. So we might want to have a look at a few cells here, on the side. Yeah. You see there seems to be no alcohol moving in here. That's why some of the cells are carried along, but it does not seem to change the structure of the cells very much. Yeah? But generally alcohol will, in high concentration, will also cause proteins to denature, which basically means proteins will lose their shape, and even if on the outside the spores might seem intact, they might still be killed off because the chemistry, so to say, on the enzymes inside the spores might actually be destroyed. Yeah. 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 See here are now some of those spores. They're definitely in alcohol here. Yeah. These here. Yeah. And that's basically what happens to it. Not a huge difference. Yeah. So could you cook an egg with alcohol? And the answer is, you won't believe it, yes. If you take raw egg and you add alcohol to it, it will turn white, just as if it is cooked. Because the alcohol will also, just like heat, cause a denaturing in the protein. Not only that, if you add some acid, some stronger acid to the egg, then it will also turn white, just as if it is boiled. Yeah. So, but if you want to try this at home, add some strong alcohol to a little bit of the egg white, and you're going to see that it will turn solid, and just as if it is cooked. Yeah. So this is because the proteins are denatured. That means the proteins will start to interlink with each other, and therefore, just like what happens when you add heat. It's kind of interesting. Yeah, you can actually cook an egg with alcohol. I still wouldn't eat it because it will taste horrible with all of the alcohol there, right? Egg and vinegar. I did not try egg and vinegar yet. I did try egg and citric acid. I added some citric acid powder. Yeah. So that's the thing. And if you want to add alcohol to egg, it's not with beer. Beer is not going to be concentrated enough. You have to go for higher concentrations. Yeah. So that's a little bit the thing here. Yeah, but that is indeed the reason why back in the day it was Joseph Lister, an important surgeon, and he actually discovered that you have to disinfect surgery utensils, surgery tools. He disinfected them with alcohol. was one of or other disinfectants, and he discovered that this actually significantly increases the survival rate of the patients. The reason is because those chemicals, when they essentially will destroy microorganisms, and they didn't know that. As a matter of fact, for many years people didn't even know that microorganisms actually cause diseases. So this is kind of so the medical history is quite fascinating, and of course people knew about spores and microorganisms, all these things. They knew about that because they had microscopes, but they did not know that they can actually be they are actually the reasons why diseases can spread. They did not make this connection, and it was actually Robert Koch, a German scientist, and also Louis Pasteur, a French scientist, who basically contribute, and others contributed significantly to the discovery of the role that microorganisms actually play. Is it okay to want to see dry sperm under the microscope? It's always wiggling every place online, but I wonder how it crystallizes. I also wonder how it crystallizes and never observed this. Let's put it this way. Many body fluids contain salt. So you take some saliva, you take some tears, you put it on a microscope slide, and when you wait for it to dry, you will see crystals. And the reason is because body fluids contain salt, and it's these salts that will actually crystallize out. So this is nothing unusual. So saliva, tears, especially tears. In saliva, there's sometimes a lot of mucus as well, but tears work quite well. Sweat as well. You take a drop of sweat, put it on a microscope slide and wait, and you will see crystals, and that is because of the salt that is included. So this is actually nothing unusual and happens with many body fluids. Okay. Do you think will ever be possible to do surgery on microbes? It would be funny if they did surgery on a grape. Let's put it this way. Surgery on microorganisms. There is something similar that can go on, and that is essentially it is possible to actually extract the DNA or inject DNA into single cells. This is possible to do that using a microscope, to actually go into a cell and put something in or take something out. If you want to call that microsurgery, well then you just call it surgery. But essentially, actually manipulating cells on that level is possible already and has already been done. I cannot do that. I don't have the equipment, but there are so-called micro-manipulators that will actually allow you to do that. Probably not bacteria, but eukaryotic cells, which are fairly large. I don't have to say that many of them die in the process. But something like this can be done. There is a book recommendation. What I would like to I recommend reading Microbehunters by Paul de Cruyff. Yes, I highly recommend this book. I've also read it, and it is a highly readable, very understandable, and I think very, very interesting book. Microbehunters by Paul de Cruyff from I think 1927. It's already 100 years old, but it is written in a very, I would say, modern way, in a very readable way. Highly interesting if you're interested in, even if you're not so much into science and if you're not familiar with a lot of scientific terminology, this is really talks about the history of all of those important discoveries in a very, approachable and accessible way. So it is a science book which is not written like a science book, it's more written like a storybook. So it's very readable, I highly recommend it. Really, and the book can still be bought. Not only that, look for it online. The text, it is already in public domain but you know what, just get the book. To have a nicely bound book is also nice to have, but the text can be found online because it's already so old, but I think the material is still highly relevant, and that's why it's still being sold. Do you teach at school in English? Yes, I teach in English. That's correct. Yes. So that is something that I do as well. So first edition is from 1926. Yes, but still, honestly, the book is I don't know, absolutely. I think absolutely phenomenal. I learned a lot from this from this book. Yeah? It talks about a lot of people, how they made the important discoveries in medicine, in microbiology, and it is actually one of those books that actually got some people, not me because I didn't know that, but it got many people already interested in studying this field at university. And people, you know what I'm going to do now? It's 1 hour and 39 minutes that I'm already talking, and I think it's time to call it quits. The spores, basically, this is how they look like. Yeah. The alcohol did not change their shape quite a lot. Okay. Oh, well, actually here, further away, the spores do look a little bit bigger. I just realized. Then maybe over here. So maybe the alcohol did take some of the water out from the spores. Yeah, I don't know. Yeah. I'm going to call it quits, people. Hope you liked it. Yeah, if you have any suggestions or anything that you want me to talk about, please feel free to contact me. Thank you for the questions, because I think questions are also very important, because they kind of also keep the live stream a little moving and going and interactive. And I'm going to simply want to wish you a nice day. I wish you all the best. Happy microbe hunting as always. And see you around next time. And do check also the videos on this channel here. I keep on uploading every now and then some videos, and you might also be interested here. All the best. Bye-bye. Hopefully see you next week again.